Hepatitis B serology is a very confusing topic. I am gonna make it very simple for you. Hello, I am Dr. Azaz from medicovisual.com. Welcome to this visual lecture. So let's suppose this is a virus infected hepatitis B virus infected hepatocyte, and this is continuously producing surface antigens, the subviral particles, hepatitis B subviral non-infectious particles, as well as the actual virus. These subviral particles, I have discussed the detail in the lecture of life cycle or replicative cycle of hepatitis B. So these surface antigens, these subviral particles, they are composed of surface antigen only, surface antigen that is embedded into envelope, that is embedded into the membrane, right? But actual virus, it not only consists of the surface antigen, but also there are inside that nucleocapsid core, and inside that is double-stranded, partially double-stranded DNA and with DNA polymerase, right? So as this infected hepatocyte, it is producing these particles, there will be rise in surface antigen. These surface antigen will be detected, right? So let's suppose this line, it is denoting the surface antigen, so you can detect surface antigen, right? And surface antigen will be positive. It is a start of acute infection of hepatitis B virus. Then what will happen? It is not only producing this viral and subviral particles, but we will also see these surface E antigen. So E antigen is being produced by this infected cell and secreted outside, as well as these virus and subviral particles are being produced. So there will be a rise in E antigen. E antigen will be positive. But E antigen, the quantity, the number of E antigen that is being produced, it is not as large as that of surface antigen. So spike of surface antigen will be larger as compared to spike of E antigen. Clear up till now? Now, can we detect this? What is this? This core, nucleocapsid core protein. Capsid protein or core protein. Can we detect it in plasma? No, we cannot. Why? Because it is enclosed, it is packed inside the envelope. It is enclosed inside the envelope. It is not lying free. So we cannot detect it in the plasma. The only way we can detect is that we have to break open this virus particle, we have to remove the envelope, and when we will remove the envelope, then we can see this core. And it is not routinely done in laboratory. It is a research procedure that through certain techniques we remove the envelope, and then we can get the nucleocapsid core, and then on that we perform different experimentations in research labs, BSL-4 labs, right? Then what happens? The only way this core protein can come into plasma, I will tell you how. It can come into plasma, but not in the detectable range. How come? Let's suppose this is another infected virus particle, and now it was producing the virus. It was going to produce the virus particle, but it has not yet completed the process. It has not yet completely assembled the virus particle, right? Now at this time, if immune cells come, cytotoxic B cell come, and it start attacking this cell, and at this time if cell is destroyed, then what will happen? This core protein, it may come into plasma, it may come into blood. It is not yet, and it is not yet enveloped. It is now naked core protein. It may come into plasma. So very little amount, because this is relatively rare occurrence, very little amount of core antigen may come into plasma, but that is below the threshold of detection. So usually in books you don't see that written, they don't write it, right? Because it is almost indetectable in laboratory procedure. Then of course all these three antigen, this is C antigen or core antigen, all these three antigen will be there in plasma, in blood. So body will generate immune response against these. How body will generate immune response against these is that body will create antibodies against these things, right? Body will create antibodies. So simultaneously body will create antibody against this core, this surface protein, as well as this E antigen, all these three things. But let's discuss it one by one. So let's suppose here, first let's suppose that body has secreted antibody against core antigen. So as soon as body synthesizes this antibody, it will bind with this antigen, right? And now as it starts binding with this core antigen, it will appear that this core antigen is decreasing. The amount of core antigen is decreasing because it is bound with antibody, and we can only detect free antigen as well as free antibody. We cannot detect bound antigen or bound antibody. So it will appear when it is binding, and let's suppose it has bound all the core antigen that is present, and when it binds all the core antigen that is present, what will happen? This core antigen will appear to be 0, right? But still we have not seen core antibody. We will only see core antibody when it is present in excess to antigen. Now whatever core antibody that will be produced, because all the antigen is now attached, so whatever that is produced, it will be now lying freely in plasma. This is free core antibody, and this will lead to a rise in level of free core antibody. So this is free core or hepatitis B core antibody. So there will be rise in anti-HBC antibody or anti-hepatitis B core antibody. So these are the things that are present in acute infection. All other are negative. Antigen, I will tell you why it is negative. Actually it is present in plasma, but it appears to be negative in lab test. I will tell you why right now. So these are the things that you find in acute infection. Now let's suppose infection starts to resolve. Let's suppose now cytotoxic T cell, they have destroyed all the infected hepatocytes. Now they are no more generating new surface protein, right? New surface protein is not being produced, and no new E antigen is also not being produced. So what will happen? The antibody against this E antigen, it will start binding with it, right? And because it will start binding with it, E antigen will appear to go down. Then what will happen? When all the E antigen has been bound and no new E antigen is coming, then what will happen? This antibody will be in excess. Anti-E antibody will be in excess and it will be detectable. So there will be rise in which antibody? E antibody. So rise in E antibody. Clear? E antigen will become negative and E antibody will become positive. At this point when infection is being resolved, you cannot detect E antigen. You can only detect E antibody. Why so? The reason is that whatever E antigen that was there, it is either bound with antibody or it has been now destroyed by our immune system. So we can only detect E antibody, right? Then along with E antibody, body is also producing anti-surface antibody against the surface protein. Again same thing will happen, it will start attaching with these surface proteins, and level of surface protein will appear to decrease. Although it is still present in plasma, and it will be gradually cleared by immune cell, but it may still be there, but its level will appear to decrease, right? But still we cannot detect the antibody unless it is present in excess. It has bound all the surface protein, and when it has bound all the surface protein, whatever anti-surface antibody is being produced, it will be there in plasma, and there will be rise in anti-surface antibody, and of course this core antibody, it will also be produced more and more. So that is the point here, where we can see that surface antibody start to appear. At this point we will say that infection is now being resolved at this point. So here there is infection is resolved. Now at this point we cannot detect, from this point onward we cannot detect any of the antigen. All of the antigen will be cleared by immune system. Either it has been cleared or it will be cleared by immune system. So no antigen will be there, only antibodies, and all the antibodies will be there. And most important antibody out of these is which antibody? Anti-surface antibody. Why so? Because it is the only antibody that is protective. If anti-surface antibody is present, it means that if a virus comes into plasma, surface antibody will quickly attach with it, and it will prevent it from entering any hepatocyte. Clear? So anti-surface antibody, whenever it is positive, it's a very good news for the patient and for you as a treating doctor, because it means that infection is starting to resolve, or it has been resolved, right? So here I will tell you an important point, that here you can see at this place, let me show you, let's remove this. Here you can see that here is the threshold of detection. Now at this point, now you cannot detect at this point surface antigen. You can also not detect at this point surface antibody. From here, from this point up to this time duration, it may be let's suppose almost 1 week duration. In this duration, where you can neither detect surface antigen, nor you can detect surface antibody, this duration is called window period. This is called window period. Actually, during this duration, I am repeating this again, during this duration both antigen as well as antibody is present, but they are undetectable because they are bound to each other. And that is why they are undetectable. When one thing is in excess, that is detectable. At this below this point, before this point, surface antigen was in excess, so it was detectable. After this point, surface antibody was in excess, so it is detectable. Whatever thing is present in excess, that will lie freely and that will become detectable. Actually, both may be present during this window period. So in this window period, we can only we can only check that whether hepatitis B is present by the presence of either core antibody or more precisely through hepatitis B PCR test. I have discussed in case of COVID-19 how PCR is done. I have discussed in another lecture. The principle is same. You can watch that video as well. So through PCR we can detect it, right? Real time PCR. Then this was about acute and resolved condition. Now what happens in chronic cases? How it becomes chronic? In chronic, what happens that body fails to produce surface antibody in excess. Why body fails to produce surface antibody in excess? The reason is that cell mediated immune response is not sufficient enough to kill all the infected cell. So whatever surface antibody that is produced, it will keep on neutralizing more and more surface antigen that are being produced. Antibody will be produced, then more surface antigens will come, then body will try to produce some more antibody, then more surface antigens will come, and in this way there will never be excess of surface antibody. There will never be extra surface antibody that is required to be protective. So surface antibody will never become detectable, only we will see surface antigen. So what happens in chronic infection is that body fails to produce surface antibody in excess. So what will happen? A little bit surface antibody, this is surface antigen, a little bit surface antibody that is produced, it will try to neutralize some of the antigen, surface antigen, and surface antigen will become a bit lower than this spike. It will become up to this level, but it does not completely go below the level of threshold. It does not become zero and antibody does not become in excess. So it will be dwindling here and there. At this point it may plateau here. That's what happen in chronic infection. So chronic infection, everything is same except this thing. So it is chronic unresolved case. So there will be surface antigen there. I will talk about E antigen later. Then there will be core antibody, and there won't be surface antibody. There won't be any detectable surface antibody in case of chronic infection, right? And now talking about E antigen, it may or may not be present in chronic infection. It may or may not be present. If it is present, it means virus is actively and aggressively replicating. It is enjoying the luxury of procreation inside the hepatocyte. It is actively replicating, and it is, of course, with each replication it is synthesizing E antigen, right? And if it is not present, what it means? If it is not present, it means virus is living inside the cell in a secret fashion. It is very slowly replicating. And whatever antigen, whatever E antigen, whatever small amount of E antigen that it produces, it will be quickly neutralized by E antibodies. So E antibodies will be positive. So again, not this, this one. Let's move this. So E antibody and E antigen, they may or may not be present. If E antigen is present, E antibody will not be there, and if E antigen is not present, then E antibody will be there. So if E antigen is present, and it means virus is actively replicating, it means when virus is actively replicating, there will be a large number of virus particles in the blood of patient. And when we remove this virus particles and when we detect the amount of DNA through PCR, DNA will be much high. Amount of DNA will be high in such case. And if it is negative, because replication is less, so amount of DNA will be very low. In such case, when it is present, E antigen is present, it means there is more likelihood of transmission to another person. This patient, this hepatitis B patient, it is more likely to transmit this infection to another susceptible person because more virus particles are present. But if it is negative, less virus particles are present, it means this person is less likely to infect another person. So E antigen is also called transmissibility factor. Presence of E antigen mean more likelihood of transmissibility. Clear up till now? Then in immunization, what we give in immunization is that we inject surface protein. We inject the surface protein of hepatitis B virus. We use recombinant protein. We inject it into a person, right? And after some time, not whole virus, only this surface antigen. And after some time, what will happen? His body will produce anti-HBs antibody, and that is immunization. Everything else will be negative, right? And when this antibody will be produced, it is very easy to understand. Whenever the virus comes, it will quickly bind with this virus and destroy the virus, and it will prevent the virus from infecting any hepatocytes. Interestingly, initially within few days, when you give a vaccine, this surface antigen, it may appear to be positive, right? Without proper history, you may be thinking that maybe this person is infected with hepatitis B. But actually, this is surface antigen is due to vaccine. Vaccine is also surface antigen. So it may be positive initially and this may be negative. But after some time, body will clear this and body will generate antibody response. So it is very easy and simple, very easy to understand that what happens in acute infection. In acute infection, surface antibody will not be there. In resolved infection, surface antibody will be there. Clear? Now another difference between acute and chronic case is type of antibody. In chronic case, IgG will be there. IgG will be there. IgG antibody. But in acute case, IgM antibody will be there. The initial antibody that the body produce is mega antibody, IgM. Why it is mega? I mean, it is a pentameric. It is a pentameric antibody, right? That's all about serology of hepatitis B virus. If you like this video, please subscribe to my channel and please give your precious comments about this lecture. Thank you so much for watching.